Genetic engineering. The best example of genetic engineering is to show you and describe to you the production of human insulin by genetically modifying bacteria, which was first done in 1978, and 4 years later it was available for public use. This was a revolutionary technique. Up until then, diabetics would have had to get their insulin from dead animals such as pigs or cows, and that didn't work nearly as well enough as human insulin, and also there was problems with supply as well as moral and religious objections to using insulin from animals such as pigs. So the solution was to try and get an animal or an organism, sorry, such as bacteria that could produce insulin in huge quantities, produce human insulin, so it would work very, very well in the human body and would have no objections from anybody in society. So the bacteria was the choice of organism here, and the particular species was E. coli. So how is it done? Well, first of all, you need to get a working gene. You need to identify and isolate that gene. Now, in all of our cells in our body, apart from our sex cells that only have half of our genome and red blood cells that don't have any nucleus whatsoever, all our cells of our body contain the gene for insulin. But most of the cells don't use it. So it's best to get the cell from the organ where insulin is actually being produced, and that organ is the pancreas. Now, once you have the cell, the task is to extract the whole genome. That's done simply. However, you don't need most of the genome. You don't need all 6 billion bases. The insulin gene is very, very small, so that has to be cut out, and it's cut out using chemicals called enzymes, or proteins called enzymes. In this particular case, these enzymes are called restriction enzymes, and they will cut out the insulin gene. Now, what would have to be done now is that you'd need to stick this gene into your organism of choice, and in this case bacteria, but it's not so easy. You need something to carry the gene into the bacteria, and that thing that carries a gene in is known as a vector, and the vector in this particular case is something called a plasmid. Let me just explain to you what a plasmid is. Plasmids are naturally occurring circular pieces of DNA found in bacteria, and geneticists or genetic engineers have been using these for a long time now. They simply cut open the plasmid and they stick the gene into it, and the bacteria take these plasmids in. So that's why a plasmid is a vector, because it is used to insert a gene into another organism, in this case a microorganism. So we stick the gene in, and to doubly make sure that that gene is going to stay stuck to the plasmid, we use another enzyme to stick that gene in. This enzyme is known as a ligase enzyme. Ligase. Now, with that gene firmly inserted into the plasmid can now be taken up by a bacterium. This only has about a 1% success rate, but that doesn't matter, because as soon as you've identified the bacteria that have the gene in, they are reproduced rapidly. So you have literally in 24 hours a culture of billions of bacteria, all containing this human insulin gene. Now that gene is then expressed, and what that means is that proteins are made from that gene by the bacteria. And you would have studied protein synthesis in another chapter, so you should recall that proteins are made by joining amino acids together to get our functional protein. And in this example, insulin, a hormone made of protein. And the bacteria will churn out huge quantities of this protein. Right, take yourself through this process again. As you watch it, just try to recall the different steps that are taking place. So I hope you recalled that the gene was identified, isolated, and cut out using restriction enzymes, or just enzymes, stuck into a vector called a plasmid, using another enzyme to stick the gene in. The plasmid is then inserted into the bacterium, and the gene is expressed to produce the protein human insulin. Now these bacteria are grown in huge fermentation vats, looking a little bit like this. So this is grown on an industrial scale for the production of human insulin that can be used as a medicine to treat diabetics. And the end result is this, something called human insulin.